Genetics is the study of individual gene function, and genomics are studies of the entire genome. Remember that the genetic information is DNA, and protons are in the nucleus and electrons circle the nucleus, always wanting to be stable. Therefore, they need 8 electrons in the outer valence shell. The transcription process for DNA utilizes the messenger RNA synthesis, and translation process uses protein synthesis. Looking at genetics, this is when we consider the copying of mutations of genes that potentially cause individuals to have various disease processes or disorders. These are the various disorders that will be noted throughout this week's discussion. And PKU is carried over from last week's lecture into this week. There are 2 primary roles for the nucleic acids RNA and DNA. First, the cells synthesize proteins by gene transcription and messenger RNA translation. Second, as the cells age and die, they are replaced by new specialized cells that are structurally and functionally the same as their precursors. DNA contains genetic information in a double-stranded molecule that can be replicated and produce identical daughter cells. DNA also provides a template for RNA transcription needed for the translation of protein synthesis. There are 4 DNA bases. 2 are larger purines, which are adenine and thymine, and 2 are smaller pyridamines, which are guanine and cytosine. DNA is a double-stranded molecule that is twisted into a helix and held together by complementary base pairing. Histones serve as an anchor for the DNA to coil around and are active in DNA replication, transcription, and translation. 46 chromosomes are necessary for the individual's genome. Body cells are divided into somatic cells and gametes. Somatic cells divide through mitosis. Sex cells divide through meiosis that occurs in 2 stages. DNA strands are compacted by coiling around the histone proteins. Greater condensing will occur with more coiling and packing, which happens after replication, but prior to mitosis. In the greatest condensed form, the short arm is the P arm and the long arm is the Q arm. The cell cycle showing both the gap and the mitosis phases. There is always a checkpoint to make sure that the cell is being replicated appropriately. The G1S checkpoint assures sufficiency of cell health, nutrient levels before the actual beginning of cell replication. Mitosis occurs in 4 phases. The first is prophase. Here the chromosomes condense and appear as paired sets or sister chromatids. The spindle fibers form attaching to the kinetochores of the chromids while the nuclear envelope disintegrates. The second phase is metaphase. Sister chromatids align around the cell equator and the spindle fiber attachment is checked. Third is anaphase. Here, the spindles pull back from the cell equator, thus the sister chromatids separate and migrate to the cell's center poles. Last is telophase. This is where the nuclear envelope reforms around each chromosome at either pole and it begins to condense into chromatin. Cytokinesis is the division where the division is completed with plasma membrane surrounding each cell. In the G1 phase, the cell becomes ready for the S phase. DNA copied similar to mitosis. Sister chromatids are adjacent and due to similarities, crossing over can happen with segments from both maternal and paternal chromosomes. During telophase 1 and cytokinesis, the new chromosomes are separated into daughter cells. The second cell division occurs for the daughter cells, reducing the chromosome number to 1 with a random chance of gametes having paternal chromosome, maternal chromosome, or mixed chromosomes. Replication occurs during the S phase. This begins when double strands separate at the origin sites. Each strand is a template for synthesis of an antiparallel strand. Here, the sequence of the new strand lines up with complementary bases to the template strand. This permits reformation of the double-stranded structure and replication ends. Proofreading during DNA replication is able to identify mismatched bases before moving further to decrease the spontaneous mutation formation. Enzymes proofread the sequence of nucleotides and upon finding an error, replace it with the correct one. The gene is the DNA template that consists of 3 parts. First is the promoter region, second is the coding region, and third is the terminator region. The promoter region instructs the RNA where to start transcription and which strand of the DNA is to be transcribed. The coding region is the portion of the gene that makes the polypeptide chain and eventually the protein. The terminator region instructs the RNA polymerase where to end the coding. Coding regions are expressed regions or exons, and non-coding regions are known as introns. During alternate splicing, different proteins are produced by splicing together different combinations of exons. After the transcription phase, the messenger RNA is then transported to the cytoplasm. Then it can begin the process of translation. In translation, this is the mechanism where the nucleotides in the messenger RNA are interpreted and produce a chain of amino acids. During the translation, the messenger RNA is read in a series of 3 codons, which are nucleotide units. The 4 RNA bases, adenine, uracil, guanine, and cytosine, can be combined in any order. Three codons are used as stop codons. These are the UAA, UAG, and the UGA. There is one codon that is the start codon. The AUG codon signals the translation process. Each amino acid has minimally one codon. Routinely, more than one codon that corresponds to the mitochondrial RNA sequence. Within the ribosome, a complementary transfer RNA carrying the appropriate amino acid will bind to the codon for the amino acid to be incorporated into the growing polypeptide chain. Because the codons for an amino acid are only different within one position, this does help reduce the chance that a mutation will change the amino acid sequence. An individual's genome is made of 2 homologous copies of an autosome plus 2 sex chromosomes. Maternal chromosomes are XX and your paternal chromosomes are X and Y. The gene locus is indicated in shorthand nomenclature, including the chromosome, the arm of the chromosome, and the gene location within the chromosome. The arm of the chromosome is the part above or below the centromere. The short arm is the P and the long arm is the Q. The remaining numbers are the region and the subband where the gene is located. There are 2 copies of each gene, one on maternal chromosome and one on the paternal chromosome. The copies may be identical or they may have a small or large difference. Each of the gene versions is an allele. An individual may be homozygous at a given gene locus with maternal and paternal alleles being identical, or the individual may be heterozygous with 2 different alleles at the locus. Alleles within the normal gene structure and function are known as the wild type. Allele versions that do not function normally are known as mutations. Remember, homozygous means the same or identical, and heterozygous means different. Gene variability or genetic variability is still being researched because not all of the functions of non-coding regions of genes are currently known. Genotype is the identified DNA sequence for an individual. Phenotype is the physical expression of a characteristic that may be altered by the genotype. Silent mutations does not cause any amino acid change in the final protein. Missense mutations cause a change in one amino acid of the protein's primary sequence. Nonsense mutations are an insertion of a premature stop codon termination protein synthesis before the full protein is complete. This blocks the cell from making a usable protein. The effect of change of a single nucleotide on the resulting amino acid sequence. The wild type is no mutation. The gene and the peptide are shown without a mutation. With the silent type, the base change from the C to a T at position 6 does not alter the amino acid sequence. The missense, the base change from a C to a T at position 6, changes the amino acid from leucine to phenylalanine. With nonsense, the base change from a C to a T at position 12 adds a stop codon and truncates the peptide prematurely. Frame shifts are an insertion of a T after position 2. This shifts the reading frame and changes all the amino acids in the peptide. This may have implication in Tay-Sachs disease, Crohn's disease, and some cancers. The deletion or insertion is a loss or an addition of a single nucleotide that changes the triple sequence. The example for this would be cystic fibrosis. Repeats of trinucleotides lengthen the gene and impact the gene product and the health of the individual. Trinucleotide repeats associated with disease often increase in number during the meiosis process or expansion. Huntington disease is caused by an expansion of the triplet CAG repeated several times in the gene sequence. The pedigree indicates an autosomal dominant pattern with two children of an affected mother also acquiring the disease. The mother's onset was age 39 and she had 50 copies of the repeat gene. The daughter had an onset at 30 years of age and 57 repeats. The granddaughter had an onset at 23 years of age with 75 repeats. This is known as genetic anticipation, with the increase in repeats and the decrease in the age of onset. Huntington disease is an adult onset, fatal neurodegenerative disease. It manifests itself ages 35 to 45. It is characterized by uncontrollable chorea, which is brief twitching, sporadic, non-purposeful movements. It progresses to increased motor dysfunction with cognitive loss and psychiatric manifestations. For Mendelian inheritance, the patterns are based on each individual having two alleles for each gene and on the strength of the genes influencing a trait. Mendelian inheritance includes autosomal dominant and autosomal recessive inheritance patterns. These relate to autosomes, which are chromosomes 1 through 22, that occur in homologous pairs. Characteristics of autosomal dominant inheritance are sexes are equally likely to be affected and to transmit the gene. An affected person will have at least one affected parent. The numbers of affected individuals in a pedigree are higher than expected for autosomal recessive and sex-linked inheritance patterns. If one parent has the disorder, there is a 50% chance of passing on the abnormal gene copy to each child. Autosomal recessive inheritance characteristics. Affected individuals are routinely born to unaffected carriers. Both sexes can be equally affected, and there are fewer affected individuals in the pedigree. If one parent has the disorder, then there is a 50% chance they will pass the abnormal gene copy to each child. The risk of the offspring and having the disorder is entirely dependent upon the affected parent's genetic contribution with each pregnancy. For genes with autosomal recessive inheritance pattern, the individuals with one copy of the mutated genes are carriers, meaning that they carry the gene and can pass it to the children. Examples of this are cystic fibrosis, PKU, sickle cell anemia, and Tay-Sachs disease. The probability of a particular phenotype being expressed in children with known genotypes of parents. The large G is the normal allele and the small g is the abnormal or the disease allele. The affected parent has the genotype large G small g for the disease gene, and the non-affected parent has the genotype large G large g. If the affected parent donates the disease allele or the small g, then the child has the gene and the disorder. Here, the non-affected individual is the large G large g. The carrier individual is large G little g, and the affected individual is small g small g. A heterozygous mother will have the phenotype of the disorder and a 50% chance of passing it to each child in A. If the father has the disorder, then there is a 50% chance of passing the disorder to the female child in B. Examples would be Rett syndrome, Fragile X syndrome, and rickets, which is vitamin D deficiency, or X-linked hypophosphatemia. If the father is affected, then all the female children will be carriers. If the mother is affected, 50% of the male children will be affected and 50% of the female children will be carriers. Examples of X-linked recessive disorders are hemophilia and Duchenne's muscular dystrophy. Penetrance is known as the percentage of individuals with a mutation who develop the related phenotype. Cystic fibrosis is 100% penetrance, meaning every individual with a mutation of the CH03 gene will have the disease. Reduced penetrance can occur in autosomal dominant and autosomal recessive disorders. Examples are retinitis pigmentosa and long QT syndrome. Expressivity is the variability in the expression of the phenotype for individuals with the same genotype. The signs and symptoms may differ in pathophysiology and severity depending on the mutations of the genes that are present. In Marfan syndrome, this equally will occur in males and females, and often occurs in every generation born. This is also the leading cause of aortic aneurysm in individuals younger than 40 years old. When considering hereditary breast and ovarian cancer, geneticists look for the BRCA genes. These are the mutated gene that are thought to cause breast cancer and ovarian cancer through genetic sharing. With coronary artery disease, we still need to consider family history. There are some single gene disorders that can increase the risk and decrease the age of onset of coronary artery disease. For major depressive disorder, it is thought to have a genetic component, and it can be familial, therefore be seen in family pedigrees. The gene's mnemonic. The G increases the probability of an underlying genetic syndrome. The E, the onset of disease at an earlier age than expected or in an uncharacteristic sex, will indicate a genetic basis. The N is neurodevelopmental delay in infants and children. The E, among these pathologies, would be finding in either groups or structures of paired or bilateral organs. Example would be finding several different cancers in the same patient simultaneously. The S example would be high lipid levels in a young, healthy patient. Karyotype is a method of aligning and marking a complete set of chromosomes. Hybridization arrays and gene chips are the current standard for cancers, some infections, looking at pharmacology and pharmacogenetics. Synthetic DNA sequences or probes are constructed as complementary to a desired group of gene transcripts. Messenger RNA from a biopsy of a tumor sample and a non-cancerous sample from the patient with cancer are isolated, then converted to complementary DNA using a reverse transcriptase enzyme and labeled. Small volumes of each set of complementary DNAs are added to each spot on the gene chip. The analysis detects transcripts that are not found in any cells, those found in all cells, and those unique to the tumor or non-tumor cells. There is still ongoing clinical and research being done in genetics. Individuals now can use products like Ancestry and determine their genetic background. This is especially important for the clinical nurse specialist due to the fact that if an individual determines that there is some genetic mutation in their background, they may have various questions. Epigenetics is another way to research human DNA samples to determine any types of mutations. For screening in children, there are various tests for genetic disorders. At 15 to 20 weeks gestation, an amniocentesis can be accomplished. At 10 to 12 weeks gestation, chorionic villus sampling can occur. At greater than 16 weeks gestation, percutaneous umbilical blood sampling can occur for the mother. For newborns, screenings are done with every baby that is born in the United States. This helps the child to live as normal a life as potentially possible given their disorder.